into the panel, Jen, if you think that that's okay.
I do. I think that would be wonderful, Sabrina. I just want to thank everyone again. Dr. Yu, that was an incredible presentation, very heart-wrenching at times in the pediatric realm. Next up for moderating the panel, which we're really excited about, is Clotilde Laguerre-Turenne. And Clotilde is an associate professor of neurology at MGH and Harvard Med School. She's also the Araminta Brock Healy Endowed Chair in ALS and the associate member at the Broad Institute. And we're really excited to have Clotilde. And there's someone else that I would like to introduce as well here. As I mentioned earlier, we're really pleased and proud and honored to have members of the ALS community here with us. We heard from Brooke this morning, and we're going to hear from someone who's who's agreed to join our panel this afternoon. Her name is Blaine Dangle. She's just like Brooke, just a delightful person who unfortunately, had her life just completely disrupted by a diagnosis of ALS. And she was diagnosed last year at the age of 38. She lives in New York City, and she's just a great person. Really excited to have her here and for her to tell her story. It's a really interesting one, and so I'd like to thank all of the panelists, the ones that could join us again for coming back, and Blaine and Clotilde, welcome.
Thank you so much, Jen. It's really a pleasure to be here, and it was an amazing day, actually. Quite impressive lineup of speakers. I really enjoyed. Maybe, Blaine, if you want to introduce yourself, we don't know each other, but I would love to hear about your journey, and then we can, what we will do. There are several questions that have been put in the chat, so I will go through some of them. There are many themes that come back, and actually Dr. Liu has summarized them very well in his last slide, so I love it as a transition to this discussion. But maybe we can start with you, Blaine.
Sure, I will try to abbreviate my story as much as possible. But before, thank you, ALS ONE, for organizing today. I'm really grateful for the experience to hear all of these speakers and actually to be on the panel with the speakers. It's kind of like, or at least for me, it's like getting to meet some of your heroes, because I think whether you know or not, a lot of you have played like a direct role in giving me so much hope and really being able to lead my life, in a normal way. And so I think, like, in terms of my diagnosis story, the interesting piece about it is that my story is maybe not as interesting as my mom's medical history. She's really been the mystery in our family for really the last 20 years. Mayo Clinic, she went to Mayo in, I think, 2003. And they had the impression that maybe she had MMA. And over the last 20 years, she's gone back to Mayo and seen other neurologists. And no one ever confirmed or updated that diagnosis, despite her continued deterioration. So I think she kind of, gave up on getting a name for the diagnosis, and was really just trying to live her life as best she could with her progression. But last year she started talking about arm weakness, and a doctor had recommended that she get carpal tunnel surgery. And at that point, my wife, who is a clinical researcher, was like, pump the brakes immediately. This sounds wrong. I think whatever she has in her legs has obviously progressed to her arms. And my wife goes sort of down a rabbit hole and starts reading all of the literature. And about a week of not talking to me because she's so engrossed in all of the just random research, she's like, I think your mom just might have sort of a rare, slow progressing variant of ALS. And so we have my mom go get a genetic test. And the genetic test comes back. My wife was right, as she most of the time is. And then a few weeks later, I am chatting with Dr. Schneider on Zoom and enrolling myself in the ATLAS study at Columbia as an asymptomatic SOD1 carrier. So I go in for my first in-person appointment, presumably to collect some baseline data for this, asymptomatic trial. And I find out during the EMG that I'm actually not, in fact, asymptomatic. And this was, pretty surprising for me. I had been having walking problems for about 5 years. And I couldn't really walk more than 100 steps a day. I couldn't do stairs reliably. I had gone to see so many orthos, so many neuros as a precaution, because I was like, maybe this is what my mom has. After seeing, 2 of the leading orthos and getting clean EMGs, I was diagnosed with flat foot syndrome. And so I had a reconstructive surgery on my right foot. And I just thought I was having a slow recovery. That might be just really powerful mental gymnastics of denial that one is able to do in this sort of a situation. But so going in, I was really unprepared for the news and sort of like blindsided, because I thought that I was just being a good citizen of the world and participating in research. But a couple things were kind of extraordinary about being diagnosed, like exactly in that moment. And one was actually that it was the day that Dr. Miller published the phase 3 clinical trial results for tofersen in the New England Journal of Medicine. It was also the day after, I think, the guy from the UK, there was like a video of this old man, and he had been in a wheelchair, and he was, walking around, and he was like, "Look, I can walk without me sticks." Sorry, I did a really bad, not British, but Irish accent there. So please forgive me for that. But, so I had sort of like 2 good data points that I was like Googling, I think, as I was getting the results. And then sort of the third extraordinary thing is, Dr. Harms and Dr. Schneider were like, "Look, you obviously can't participate in this trial since you're not, asymptomatic or pre-symptomatic, but we'll take you under our clinical care." And like 4 weeks later, I was getting my first tofersen injection through the EAP. This morning, I had my 14th tofersen injection. And that patient, the last patient that Dr. Miller spoke about in his talk, the one that can now shower without the shower chair, that's me. I hope I'm not breaking any HIPAA rules by adding myself, but since this is my data, I think it's probably okay. So that is still true. I can now still shower without my chair, although for those of you who haven't tried it, showering with a shower chair is pretty luxurious. So I'm in a constant battle with my wife about where the shower chair lives in the shower or not. But really, I'm like able to do all of these things that I haven't been able to do in 4 years. And every month I'm actually still gaining function and strength. And these things, they sort of seem small, I think, to the average person, but they're all adding up and giving me this life where, and a lot of, like most days, I don't actually think about the fact that I have ALS. Dr. Harms wouldn't even give me a permanent handicap parking pass after seeing my strength score improvement. And, honestly, like most days, I just think about how cool science is and how grateful I am and how much hope and how much I believe that this is really just like the beginning of a new era for treatments for this disease, thanks to the work that you all are doing. So thank you.
Thank you so much for sharing your experience. It's very nice to hear from you. I think, everyone here will agree with me that the day was, I don't know, for me it was very exciting. I'm in the field, so I know we're making progress as a field, but to see that within a day with so many talks, with so many advances that actually are now impacting patients, I think that's a change. Compared to many of the meetings we are used to have, and it's really nice. This morning we have heard Dr. Babu, Dr. Cudkowicz showing how much innovation there is in the development of the clinical trial, and I'm sure there are a few questions that have not been addressed, and I will ask. But I think also to see how genetic approaches, in a large term, as Dr. Watts has demonstrated, like there's so many such variety in the approaches that we have now and in the tools, and to see that they're really moving to clinic, it's incredible. So I'm going to take a few questions that have not been addressed, and first one is really related to the innovations in trials that has been discussed this morning by Dr. Babu and Dr. Cudkowicz, and the question was whether there's any statistical innovation that accommodate for more heterogeneous populations, yet keep trials short and durable. And that comes back also to the question of accessibility to the trial of more patients, and also including patients who are maybe more advanced in the disease. So I don't know if Dr. Babu or Dr. Cudkowicz or anyone else.
I'm happy to say a few words and then get Suma's input. So yes, absolutely. So there are now ways in your analysis to account for that heterogeneity. And the best one really out there is this TriCal score, which is a score developed by our colleagues in Europe that takes certain baseline characteristics and kind of gives a score that has some predictability about that variability in that clinical course. And so we've now included that in the statistics as a variable that we can stratify in analysis. So that's one of them. Obviously now there's also neurofilament, and there'll be some other fluid markers that we can use to account for that variability, allowing us to take broader participants. There's also, the option to go longer. It's always the balance of, if you take people more heterogeneity, you might have to go longer or larger trials versus the shorter trials with more homogeneous population. But, I really think strongly we need more than one way to do trials, and really got to tailor it based on the therapy and how long you think it will take and which population might work best in.
Merit, I completely agree. And I would just add to that also that you're absolutely right. There is a trade-off sometimes, whether we want to do longer trials, bigger trials, more homogeneous, more heterogeneous. We also have this opportunity now through the NIH-funded Act for ALS EAPs, where we can enroll or develop, design, and run expanded access studies in parallel to an ongoing clinical efficacy study to get real-world data and not just provide access, but also conduct research using that data set too.
That's great. So actually, maybe you've already answered the other questions that I had, but which was really how long, the trials should be, designed. And that comes really to different comments from both Dr. Miller and Dr. Schneider that it takes time to heal. And at the same time, Dr. Babu, this morning you were telling us, we have a bottleneck of not too many, but so many trials coming to the field, and that there was some really great interest in trying to designing trials that are shorter with a lower number of people. And these two questions are really entangled, right? So are there any way that we can predict for a specific modality which type of trial we would need? And Merit, you just said you think we need some flexibility, but how do we, predict which one we should do? So maybe for this, do we think that antisense oligonucleotide would require longer trial? And do we know that or not, right, based on what has been said this morning?
Yeah, so my good friend Jeremy isn't here, but I can repeat what he always says, is that the tofersen trial wasn't too short. It was too small for 6 months effect, which we only really knew after the fact. That if, we did start to see a separation at 6 months, it's just that the sample size was short. So, but we got the benefit of having that blinded crossover into the open label extension where you could see the effects later in that sample size, but we could have seen it if we had that bigger sample size earlier on. So I think it does depend on your drug and your preclinical data, telling you maybe how long it takes for your drug to work, and having this option to have these open label extensions can really help you kind of get the best of both worlds of not keep people on placebo too long, but also be able to see the long-term effects.
So maybe a question related that was put in the chat from Dr. Brown to Dr. Schneider, and it was, I don't know, Bob, if you want to ask this question, but it was about the significance of the correlation between the respiratory downturn at 425 weeks in your patient and the inflection point in her course, meaning the beginning of the rise in her ALS-FRS.
Yeah, in other words, it was very striking that the clinical trajectory began to go up exactly at that point, if I read your graph correctly, at which the patient had this respiratory crisis of some sort. And is that just chance? Was there some intervention in management of the respiratory status that permitted the infection?
Right. So you know, this is an N of 1, of course. I can't really draw any, I think, broad conclusions here, but you know, I think part of her ALS-FRS decline was not exactly related to her disease, not directly related to ALS. I mean, she was getting sicker, and she was losing function, perhaps not as a direct consequence of her motor neuron function, and you know, I think except for that, maybe it wouldn't have dipped quite as low as it did, and maybe some of the initial recovery was just because she was in the hospital. She was getting a lot of attention and care, and initially when we began to see the improvements in FRS, I didn't attribute it to the treatment. I thought that she was just, you know, getting very good attentive care. She was in the hospital for a while. She was in an acute rehab facility after, but then it continued, and so and continued steadily to you know, a degree that I think can't be explained by anything else but a motor recovery, and something directly relevant to the underlying motor neuron disease. So you know, I don't you know exactly how to interpret that inflection point. I think if not for some of the medical issues, maybe again it would have been more of a subtle plateau and then increase in time without the very sharp inflection. But this is just of course speculation. What's important here, I think, is that it's an example, one example, and I hope there are many more soon, of what improvement looks like. And as we get more of these, we will be able, I think, to draw broader conclusions. Not everybody who's getting the FUS-ASO is as sick as Anna was. Some were sicker. And I think that somebody's trajectory is going to be very different. Blaine's, if we drew it out, wouldn't have the big swings that Anna did, but it's nonetheless, you know, significant. And, you know, to the point of when, you know, how to anticipate the timing of this response, it's going to depend. It's going to depend on the individual case. It's going to depend on where people are in their disease course. Are you getting to them early, later? I think this is true of the genetic forms of the disease and and even the sporadic forms of the disease. You know, if there's more accumulated damage to the nervous system, it's going to take longer to see the plateau in the functional recovery. If someone, as thankfully in Blaine's case, who has a much, you know, more protracted course, and has lost less function, you know, her declines won't be as dramatic. She might not make it into Der Spiegel, but you know, it's for her life, it's very significant.
May I just pick up, though, if I may briefly, on an earlier point about the length of the study? So I greatly admire the Healy platform study. Sabrina, Merit done an incredible job with that. But I think there's a real risk in drawing a conclusion at 6 months. Rather, I think that is fine for initiation of crossover, but I worry a lot that we throw the baby out with the bathwater if we draw the conclusion at 6 months. And we've seen that with tofersen. We've seen that with Amylyx, and here's Anna. The case of Anna would actually also issue the same caution.
I agree.
I think it depends on the drug. I mean, I don't mean I would never disagree with my mentor, but I don't think we can say those kind of statements. And I hope the field is moving the opposite direction, right? As we get better biomarkers, kind of like again like oncology and MS, they start out with the long trials, but then they develop the tools so that you can predict clinical response in a shorter time. But you know, we're going to be a lot smarter in about 4 months because we will have repeat studies of Edaravone and repeat studies of Relyvrio that are longer and bigger. But again, they could show different results because they're bigger. I mean, there's two variables that are changing: small studies and short studies to long studies and big studies.
Great. So I mean, I think we are all very much excited by this observation of improvement in patients. I have to say that in the preclinical field, we have discussed that for many years, and we could see that in mouse models, which was very encouraging. But now knowing that it's actually happening, I know it's only a few cases, but I think it's really support what Dr. Miller was describing this morning about maybe the motor neurons being able to resprout and re-innervate muscles. There was a question that Dr. Miller has to some extent answered, but I wanted to ask it again to see if there are other ideas, which was from Dr. Werner, asking whether, so how can we tell the difference between a significantly impaired neuron axon and a dead neuron? Both have lost function, but the former might be rescued. And so Dr. Miller, was saying it's true. We have no ways at this time. Are there any ideas from this panel? I mean, I was wondering, and it might be very naive, but what about nerve biopsy? Could we see if the axons is already retracted or dead, or is there any, any examination that could be done or could be thought about? To apply in patients to answer this?
I mean, one possibility is, as Tim suggested, was that axons that are under attack, motor neurons that are under attack, are rescued by the ASO or whatever therapy, and sprouting is encouraged. There, you would see the same number of units, but they would be larger units and a more robust, response. The CMAPs would go up as he showed. And the other possibility is that there are axons that are beginning to withdraw from neurons under attack, and those units are no longer counted, but that those too are able to be rescued, and maybe part of the delay, Anna's delay and other delays that we might see, it means that the axons reverse their retraction and then reinvade the muscle and reinnervate vacated NMJs, and then you would see an increase in, I think, in the number of units as well as the size of the CMAP. So, I mean, I think now that we begin to know what these successes look like. I wish I had done this, from the start in the expanded access program. We're going to hopefully do it as part of Silence, but the idea would be to get quantitative measures of these units and then see what's actually happening physiologically. I don't think a biopsy is possible, or even necessary. So, but these are the kinds of questions that I think we'll begin to ask in the context of success with tofersen, with the FOS-ASO, and others hopefully coming soon.
So we have already discussed now the length of trial, but there is also the question of when, and several speakers have mentioned, that early treating early should be beneficial, and also preventive trials have been mentioned by several speakers. There was a comment from Jan Swindler in the chat saying FDA has been clear. They see the pre-symptomatic stage as eligible for clinical trial, as evidenced in our patient listening session transcript. And there's a question from Dr. Mail to Dr. Schneider, which is also related to that, where he's asking whether we will require another long clinical trial before iron 363 can be offered to pre-symptomatic first mutation carriers. Do you have an idea as to how large the pre-symptomatic first mutation carrier population is? So it's a large question, but maybe we can answer this precise one. And anyone who want to comment on pre-symptomatic trials in pre-symptomatic is welcome.
Well, far be it for me to predict what the FDA is going to do in these situations, but to Dr. Mandel's point about the bronze, silver, and gold, I think bronze is treating symptomatic individuals at whatever stage they come to you. Silver is treating the symptomatic, the non-symptomatics who have phenotypically converted, EMG changes, a biomarker elevation, something that signals the onset of disease. Atlas hopefully will prove that, others, too. And gold, in my mind, is pre-symptomatic, pre-onset treatment. And, Clotilde, you talk about the preclinical data. I mean, this informs us, right? There's theoretically no reason why the prevention of the phenotype that we see in the FUS mice can't be achieved in a human being. The only question is, is it safe to treat people for, as somebody, I think, Bob said, 20 years, or more, before predicted onset. I would, so that's going to be a very much of a gene and target specific, question and answer. But, it may not be that you have to achieve the same degree of knockdown with these ASOs in an asymptomatic, pre-symptomatic individual as you do in a symptomatic. You're asking the therapeutic to do a lot less. These are dose-dependent phenomena. And if you could reduce the total concentration of a protein by 50%, well below the saturation concentration of these proteins, you may have, a very significant prophylactic benefit that wouldn't necessarily be the same, in terms of efficacy as in the symptomatic treatments. And I think Jean made a point in the comment I saw about giving up too early, right? I mean, I think it's easier probably to achieve success in the pre-symptomatic population than it is in the symptomatic population. And we need to bring the FDA along slowly from bronze to gold in this. And my hope is that they will come along with us, see the power of this argument, preclinical data driven largely, and not make us do these trials, which frankly I think are just impossible to do well. There's going to have to be a leap of faith at some point.
Any other comments about treatment in pre-symptomatic? Or?
Yeah, I'd like to make a comment, just in terms of our experience. If I can get on site here. My video will stay on. It won't stay on. It doesn't matter anymore.
Okay, we can hear you very well.
Yeah. Just in concert with what Neil said, a couple of points of interest. When we first went to the FDA about SMA for our pre-IND, they were insistent that we do type 2 SMA and not tackle SMA type 1. And the reason I say that's in concert with what Neil said is that we went back and we said we don't want to treat type 2s because we feel that, and we know the disease well enough, that there's a very rapid loss of neurons. And in that rapid loss of neurons, we won't have any possible opportunity to treat pre-symptomatic neurons before they die, basically. And we finally convinced them, and that's when we got to treat the type 1 patients. And what we learned is that if we can treat pre-symptomatically, we may have neurons that have been affected and been what we would call in a phase of illness, a sick state, before we could treat them and save the neurons. So that we convince them to treat pre-symptomatically in type 1s after identification of the genetic defect, and that was where the big efficacy window came in, and how you analogize that to to ALS, I really don't know. I mean, can you nearly, if you have a clear target for treatment, then you can do that, and then if you can show efficacy, but you have to have a clear target. I don't know what the target is for ALS, if there's an analogy for the SMA population to treat pre-symptomatically. So yes, if you can treat those pre-symptomatically, you do have a great opportunity. If you know what the target is, as we did in SMA, and the target was 2 years, that's not an unreasonable window where you could see gradual improvement over time in the patients. It was perhaps a different scene than you might see in ALS. If there is an analogy to ALS, then I would go for it. But I don't know whether there is, and maybe you could comment on that, whether there is opportunity that would analogize the treatment in ALS.
Anyone would want to comment on that? I mean, I think, Bob.
Yeah, well, I mean, it's an obvious point, I guess, but it's worth emphasizing in this context, if I understand Jerry's question, that every study that's been done in the SOD mice suggests that to treat earlier is clearly better. We've seen that with the virus, with the synthetic microRNA, we've seen that with the DICI. So that's a clear pattern, and I suspect it probably is generalizable to humans. So part of a response anyway, Jerry.
Actually, Bob, now that you are discussing with Dr. Mandel, maybe you had two questions about immunosuppression. One was about prednisone, and I see you have put another one. Do you want to ask these questions?
Sure, thank you very much. Yeah, I think one of the issues, obviously, one has to deal with the AAV, as I understand it, the AAV9 story is, as you've said, that select group of patients who have pre-existing antibodies, and it would be amazing if pheresis, in fact, allows you to treat them. But I guess I'm wondering about whether or not you expect there would also be a longer-term cell-based reactivity to the capsid that might produce problems down the line. And then that relates to the second question, which is, I think, important, which is possible retreatment of a patient once treated with AAV9. And what would be required? I mean, presumably there's cellular as well as antibody-mediated reactivity that one would have to contend with. So I'm just curious what your thoughts are about that.
I agree with that you would have to consider in considering long-term treatment, you would have to consider cell-based as well as antibody, and that heightens the challenge. So beyond pheresis, you'd have to go to whether you'd go to a long-term sirolimus, tacrolimus. Tacrolimus is really B cell, but sirolimus and maybe cyclosporin or other means of knocking down T cells. But it is a different setting than than pre-existing antibody. Pre-existing antibody, I think, is an easier target. We did show that was effective in non-human primates with AAV8 antibody. We did have success in getting good gene expression in the non-human primate. So I think there are two different scenarios, but one would lead to the other. I think you'd have to have both. So the demonstration of pre-existing antibody will be important, but it's not the whole story as you suggest.
Thank you. But may I ask a quick follow-up? And again, I'm certainly not an immunologist, but there is some contention that using more than just prednisone for a few weeks, 3 to 6 months after the initial AAV infusion might prevent subsequent cellular-mediated immune problems. I don't know if there's enough data yet to draw any conclusions about that, but I'd love to hear your view of that.
Well, I think it's if the patients don't have pre-existing immunity and we protect them in the initial gene exposure, AAV exposure, we do get we do unequivocally get long-term gene expression. And so I think we can feel fairly confident. I mean, nothing is likely to predict the outcome of every patient, but I think the majority of patients will benefit from that initial immune suppression, and that's basically what we've seen. We have gene expression out in the ALS patients almost 10 years now, and so it's pretty convincing. And in the Duchenne patients out to 6 years, treating the motor neuron, I always wondered, in thinking about, we've had really an easier time with treating the SMA patient. We've got one neuron, one nucleus that we can that we can transduce, and if we can treat that before the neuron is overwhelmingly destined to die, then we have a very good outcome, and that's basically what we've seen. Mm-hmm. I said going from starting bronze, silver, gold, maybe there's not exactly the same opportunity in ALS because it's much more difficult. Although in ALS, I think from what I know of ALS, it's a much more chronic disease than SMA. So that treating patients who are symptomatic and showing that you can stop progression is a little bit more analogous to what we see with an intramuscular injection in a slowly progressive muscular dystrophy. And we've seen that with limb-girdle too. We have patients who are symptomatic with limb-girdle dystrophy who are having very good outcomes now with systemic delivery.
Great, thank you so much. So I think we should also discuss really the promise and the challenges of therapeutic development in these extremely rare cases and what we call N of 1, right? So Dr. Liu and Dr. Schneider and others have discussed that today. I think Dr. Liu, your last slide was really saying, how do we go beyond anecdotal description of what we see and what are the challenges of that, and do you want to comment a bit more on that? And there is also a question that just appeared in the chat, which was, how do you fairly select the first patient for an N of 1 or compassionate use program when more than one patient exists? So do you want to comment on these challenges?
I'll take the second question first, and then touch on the first, and then invite my panelists to comment. I think that when there are multiple patients that exist, and selecting a first patient is an incredibly awkward affair, and it's pretty fraught. One has to consider that not all patients with the same genetic diagnosis and the same clinical presentation are, of course, alike. And there may be differences in the genotype and the predicted severity of the condition. There may be critical differences in the stage of the disease, and also differences in their willingness and their assessment of risk and quality of life. There may be critical differences that might separate a subset of patients from a population from others. But there are many conditions where you can't distinguish. There are like patients. And one of the general principles that we try to adhere to is like patients should be treated alike. And so offering something to one that is really not indistinguishable from another is not easily possible. In our cases, we haven't had to wrestle with that. Mullison was a unique mutation. The adapexin mutation was a unique mutation at the time. It turned into 2, and then it turned into 5 more. But at these points now, we have systems for managing that, for first in human studies and trial registration and trial participation. There are articles written about how best to manage this type of situation. Even our KCNT1 epilepsy trial, we worked with 2 individuals with the most severe genotype and the most severe clinical presentation that distinguished them from other children who had milder versions and milder, less predictive genotypes. But I think that this is, especially in the ALS field, something to really think about and to get right. I'm sure that colleagues have more to say about that. On the first part of the question, maybe I'll come back to that later.
I would say, in terms of the question of whom to treat first, we're operating in Silence ALS on a sort of first come, first serve. We started very small. We had a handful of patients. They were coming, directly out of our clinic. But I think it's important to realize, a few things. In our situation, many of these are individualized ASOs. These ASOs don't target a specific mutation. They target a SNP that is linked to the mutation, and so it means that 2 patients with the exact same mutation, even family members, may not benefit from the same ASO. So, and there are technical challenges to each program. One patient with a given mutation could have a single SNP, let's say, that as a potential target. Others might have many, depending on their haplotype. And so the technical challenges are going to be very different, and the time it's going to take and the potential success or failure of the program will vary from case to case. So the first patient you accept for an ASO development program isn't necessarily the first person you're going to be able to bring into the that ASO into the clinic, treat in the clinic. So I think as we grow the infrastructure to do these kinds of things, this will be more of a problem. We're going to have to make some choices about given limited resources. They'll always be limited, I think, given the need here, as to whom to treat, whom to prioritize. We do follow our patients longitudinally in the year plus that it takes to develop an ASO, and people can progress out. It could be that they become so affected that it doesn't make sense, given limited resources, to move forward into the clinic with an ASO when there's somebody else who is more slowly progressive and could potentially benefit. And these are very difficult decisions, and setting up the structures that Tim is and Lorem is and others, to deal with this and scale is really important part of what we're doing now as we grow.
So, Dr. Liu, did you want to make another comment? I see the time. Are we strict on time, Jen, or can we continue the discussion? There are so many questions.
I think we're fine to continue as long as everybody can stay on. I think we're perfect. Yeah. Thank you.
Dr. Liu, I think you wanted to make another comment, but otherwise we can.
Oh yeah, I didn't want to neglect your first question about whether what's the value of ends of ones or ends of twos or ends of fives. And of course, the evidentiary value is obviously goes up hugely the more patients that we can treat. But I also, and I think that that's a scientific and statistical fact. I think, however, that I don't want that to relegate the ends of ones that are potentially amenable to these sorts of interventions to obscurity. And I think that there are scientific solutions where you can extract more from less. And I think that part of that comes from being able to rely on biomarker data, as Neil and Bob and John have so elegantly done in their small N trials as well as the large N trials, looking at neurofilament responses, even imperfect as it is. I think that looking for other generalizable ways to extract signals of target engagement from different heterogeneous diseases. Wouldn't it be wonderful if there were a generalizable approach to look at the levels of any arbitrary gene expression from the CSF of a patient being trialed on an investigational ASO? And if one could pull exosomes out from the CSF and look at target engagement or levels of either the direct gene target or some clinically relevant biomarker, so that you could again maximize the scientific evidentiary value from those in a way. These are those are, while the clinical responses on N of one basis, one worries that they will at some level you can't escape that they will be anecdotal. But these sorts of common approaches that can be applied to multiple Ns of ones can be critical.
I think a question I wanted to ask, I mean, you and others who have developed ASOs for these rare forms, but more generally, right? I think we agree there are so many targets now that we could, approach using ASOs or the variant siRNA that Dr. Watts has described. In particular, Dr. Watts, you mentioned, the possibility of targeting pathways rather than genetic forms, and so basically the possibilities are immense. But that said, I am struggling with something that I had not realized. I have worked with Ionis Pharmaceutical for many years through my collaboration with Don Cleveland and with Frank Bennett, but I realized in the past few years that actually a lot of the ASOs that work in vitro in iPS neurons, for example, actually don't work in vivo as soon as we put them in mice or non-human primate. And it's to the order, for what Ionis tells us, of 70% of the ASOs that are efficient. And this is really something we tested with the StatMin2 program, where we had to build a StatMin2 humanized mouse because we could not do that just in the models that were available. And that is really a challenge, right? Because if we have so many targets and we have so many programs, what are the, what is a pipeline that we all agree is necessary to move an ASOs from preclinical to clinical? And I would like really to hear your thoughts about that, because I don't think it's established at this point. I think there's been many different ways.
But who would you like to take first
So
attempt?
I think, any of you who have moved ASOs from preclinical to clinical trial, do you want to test? So everyone is testing the safety of the ASOs, or at least the potential toxicity, in animals. But there are several programs who have gone to patients without, demonstrating therapeutic effect in animals. And I'm really surprised by the fact that, Ionis is telling us, even if an ASO works very well in vitro, each sequence is very different. So I think this is the difficulty with the ASOs or the siRNA, that we think about them sometimes as the same drug, but it's actually, each one is very much different, as its own, potential toxicity, but also pharmacodistribution, and just one base pair makes a huge difference. And of course, also chemistry makes a huge difference too. And so like, what are your thoughts on establishing some, some recommendation, I think, for the field on how do we move as fast as possible to patients, including in very rare cases where we don't have animal models, right? How do we do that?
So maybe I'll start, Tim, unless you wanted to push. I mean, I think that you're hitting on a bit of a controversial subject here, and one that's not been resolved. Yes, it's relatively easy to develop an ASO to use in our mice, and to use in the lab. It's another thing entirely to build an ASO as a drug, and there isn't, there's guidelines that Tim reviewed from the FDA based on his experience and mine and Bob's and others, and that requires a still, to me, surprisingly small amount of safety data, to go into the clinic, and that is based on a comfort that the FDA has with this very specific chemistry, in this very specific modality, and the guidelines do not apply. The ASO guidelines do not apply to the siRNAs that Bob described, or other chemistries, other modalities. This is a unique program for the ASO, based on prior experience. But most of that experience, and this is maybe my bias, comes from Ionis and now and Lorem, but there are many other groups that are capable of building these ASOs, and they have their own pipelines that are distinct from the Ionis and Lorem pipeline. Are they the same? Are they different? Are they as good? I mean, there's a real need, I think, I agree with you, to set guidelines here, because, if things go badly in any one ASO program, no matter what its source, that is going to, I think, have implications for all of us working in this ASO space. And it's also important to note this is not a program that's going to apply to targeting pathways, right? You're not going to be able to do this with a Stathman-2 or an UNC13A, pathway that you believe is involved in the vast majority of ALS cases. We're talking about ultra rare cases here. But I would also say that the ultra rare has real relevance, and just to speak to some of Tim's points about the learnings. I mean, the learnings happen across programs on a sort of operational and practical way. But in the ALS field, as we do more and more of these, Bob's C9 studies, the SOD1 studies, the FUS studies, the stuff's coming through with, all of these inform one another, right? And which makes, I should just say, a point for importance of data sharing. But we're talking about the rare cases, and I think there needs to be some agreement, and maybe this is something Tim's working on as part of the N=1 collaborative, as to what in the end the FDA is going to tell us what's okay and what's not okay, and I think we want to determine what the pipeline looks like before they do, after a problem.
Well, I mean, one of the questions is the pipeline similar or different for the N=1 and for the other examples you have mentioned, Stathmin-2, Ataxin-2, whatever, right? That are or C9 or 72 that are applicable to a larger population. I don't think there are really guidelines on what is a preclinical package at this point. And there is safety, and as I said, there is also efficacy. And the efficacy is not, necessarily demonstrated in vivo, because in many cases we don't have the models. But yeah, anyone wants to comment otherwise on this?
Let me add one more layer real quick, and then Tim should add too. I think your point is really well taken, Clotilde. We see the same thing that Ionis sees, where we have compounds that succeed in cells and don't succeed in mice. But the other layer that I want to add is that even when we have target engagement efficacy in an animal model, that doesn't necessarily guarantee clinical efficacy in a human. And so if we can be confident that our drug is safe, which we can do in wild type animals where there's no model available, in a situation where the risk of not treating is higher than the risk of treating, I think there's a case to be made that sometimes it's right to proceed to a clinical trial carefully, safely, even without the ability to get animal efficacy models in place. Just because it's a smaller risk that the drug fails if we don't have animal efficacy, but there's also, still risks and significant time delays in some cases, especially when thinking about the complex RNA biology in a human neuron and potentially in things like target combinations. Those are just really hard to model in animals. And then one more quick layer is around the potential that some of the platform level problems in translating might be solvable through data sharing, and particularly through letting machine learning algorithms look at large numbers of ASOs that have succeeded in cells and failed in vivo, for example. If we can share that kind of data, we may learn as a community how to avoid some of these failures. So Tim's work with the N of One Collaborative, our work with the N of One Collaborative, and others are, hopefully going to do some of that, and I know others are working on it too.
The numbers of patients that we've treated and the approval of products using the PMO, the phosphorodiamidate oligo, has been very, very safe, and I would think you could extrapolate from that to the targets for ALS. The efficacy, I would say, is in the bronze category, probably because gene expression is relatively low, but the safety factor and it's used now in the muscular dystrophy community for treating patients who have pre-existing antibody, who have no other means of getting gene therapy. So it's used as maybe you can consider as a pharmacologic agent that puts patients on hold, stops them from progressing, but the numbers are incredibly high, and the safety factor is very, very powerful. It's hardly any side effects.
I'll add a comment too. I think that I think at the heart of the matter is that we're talking about how to manage risk. And one thing that has come out explicitly, first of all, I think it's very clear that none of these platforms are perfect, and none of them are simply programmable. They all have their critical idiosyncrasies, and certain sequences are not tolerated, and certain sequences are unaffected, are ineffective. And yet, how do you proceed with an imperfect platform? And when we think about risk to the patient. I think maybe one of the critical, uncomfortable things to discuss at the heart of some controversies in the field are risks to how do we weigh risks to patients in the setting of irreversible and progressive and fatal diseases, and also how do we weigh risks to platforms? I think that platform risk is something that patients may not care about, probably don't need to care about, but the industry and the field and companies especially are incentivized to care about. And sometimes the two of them are in opposition. And I think one of the critical things to wrestle with is how do we navigate this tension that from a patient clinical risk perspective, I would love if we could negotiate a happy compromise where we have a system that is resilient to failure, a system that is resilient to inevitable learnings that will come from treating patients. Fyodor Urnov often likes to say the only way to learn about and to advance the treatment, to advance genetically targeted interventions in humans is to perform genetically targeted interventions in humans. And I think that I would love if we could negotiate some balance where we're able to build a system that isn't afraid of taking appropriate and thoughtfully accepted risks. Bad risks cannot be accepted, but clinically thoughtful risks shouldn't have to sink the whole boat.
Great, thank you. I mean, there are so many questions, but I also see the time. I don't know, Blair, are you still online? Because there was a question for you. I wanted just maybe to finish with this one, but I don't see you anymore. Do you see Blair? No, maybe she had to talk about. Okay, good. I mean, I think, again, it was really an amazing day. And thank you so much for the discussions and answering. Anyone, if you did not have the answer to your question, I'm sure you can contact the, the investigators and continue the discussion. I'm sorry we could not go through all of them. Jen, will you say?
Sure, absolutely. I just didn't know if there were any final thoughts or final questions, but honestly, as I sit here, I just have so much overwhelming gratitude for everyone who took part. Clearly, what you are all doing with your time is so super important and is just making such pivotal, huge advancements. And I feel a little funny about having this conference and taking you all away. But then again, on the flip side of that coin, there's so much to be said for the sharing of this information. And honestly, to have the lineup of these experts, all of you in one place is pretty incredible. And we're very, very lucky. And we're truly, truly honored. Just sending my overwhelming gratitude again to Drs. Babu, Barry, Brown, Dr. Cudkowicz, Lager-Toren, Drs. Miller, Mendel, Paganoni, Schneider, Watts, and you. I can't thank you enough for your time. Also would like to thank Dr. Shel Lalji and her husband, Dr. Leigh Hochberg, for investing, in a student scholar, and congratulating Dylan Blue again. Thank you, Dylan, for taking on ALS and working in that field. I think we've talked about earlier today, collectively as a society, we need more people working on this disease. And so congratulations and thank you for all of your hard work there. Also to the incredibly inspiring women who we had on today, Brooke Eby and Blaine Dangle, just your stories of resilience amid, the backdrop of such darkness and heartache is just incredible. And, your success stories, we just pray that they continue that way. And to everyone who has joined us from around the globe, I look at the names on here and there's people from all countries, all walks of life. There's families newly diagnosed, people who have been traveling along this journey for a long time. And there's people in the medical industry. There's researchers, scientists, and just so many folks coming together. And this is really just what helps to move mountains. And we know how big of a mountain ALS is. So thank you all so very much. Thank you to our sponsors. And we hope you can all come back tomorrow after a good night's rest and join for industry. It will promise to be another engaging day as well. So thank you so very much. I hope you have a great night.
Thank you.